Okay, can you hear me? Okay. So, thank you all for being here and for the organizers for inviting me to speak in this symposium. The work I'm presenting today is part of my PhD thesis under supervision of Dr. Lindy, and it's in collaboration with David and Ryan at the University of Arizona and with Claudia in Spain. So, as you know, there are different types of mutations, and for example, we have substitutions, insertions, deletions, etc. The ones I'm interested in are single base substitutions because they tend to occur more often. There are different ways to look at them and classify them. One of the ways is the simple transition transversion distinction. So this way we will have two types of mutations, and then we can create a spectrum out of that for any mutational data set that we have. Or we can go in more details and consider every one single base substitution as one type. So an A to G will be different from a G to A. They will not be grouped in one type as before. And this gives us more information, let's say. But we can even go deeper to what we call the 3-mer level, and that takes the context into account. So for instance, a C to A mutation is different when the C comes between an A and a G as compared with when it comes between an A and a C, and so on. And these different contexts tend to have different mutation rates in different spectra or different samples. And so this gives us even more information and a bigger spectrum to compare and to learn from. And this if you consider all possible contexts, you get 96 different types of mutations in this case. Okay, so this is a fictional example. On the y-axis, you see the 6 different possible types of mutations, and on the x-axis, and then on the y-axis is the counts in a given sample. If this spectrum was not biased, then all the bars will have the same height. But this spectrum is biased because these two types of mutations are oversampled compared to these rare mutations. And so an organism can get access to these rare mutations using 2 different mechanisms, or maybe there are more. But the ones we studied were either an increase in the overall mutation rate. This will give more sampling of these previously overrepresented classes anyway, but more importantly, it will give access to these rare mutations, which can be good for an adaptive population. The other way of accessing these rare mutations is by reversing the bias. So undersampling the previously oversampled mutations and oversampling the previously undersampled mutations, like in this case. So the interaction between these 2 different changes, we studied theoretically in this previously published paper. This is the QR code for it if you're interested. But it applies to any asexual organism. And so I'm not going to talk about it. I'm going to talk about my next project, which is an application of this idea in cancer. And that's why I'm in this symposium, because cancer is within organism conflict, right? So a cancer driver mutation is beneficial from the selfish cell viewpoint because it increases its fitness. It allows it to outgrow normal cells, but it can lead to the death of the whole organism, right? So the question here was, cancer driver mutations are rare in normal cells. So were they accessed by cancer cells simply because cancer has higher mutation rate, or is there also a change in the bias that enhanced this access? So we worked on cancer genomics data. We analyzed data from BCOG dataset. We have data from 20 different tissues, as you can see here. Some of the samples are hypermutated. These are the ones shown in red. These have more than 10 mutations per megabase, and the others we call non-hypermutators, okay? And so I'm going to use these cartoons throughout a little bit. So happy faces in yellow will be for normal cells, evil faces for cancer, and in blue for non-hypermutators, and in red for hypermutators, just to help you like follow and keep consistent with the colors. So the first question was, where is it? Okay, so the first question was, how similar are the trimer spectra between the different samples in different tissues? Between the different tissues. And so, for instance, we took the trimer spectrum in bladder and correlated that with the spectrum from all the pooled mutations in all non-hypermutated samples in the other tissues. And we repeated that for blood, for bone, for brain non-hypermutated samples, and so on. And then we repeated that for the tissues that have hypermutated samples. And we saw high correlations in non-hypermutated samples, which means that different tissues tend to have very similar spectra when there's not a very high mutation rate, as opposed to hypermutators, which show lower correlations, so the spectra are less similar. So this comes to our question. Okay, now is there also a difference in the biases between these two sets? So back to this example. So our goal is to test whether there's a bias spectrum change in cancer, but what are we going to compare with? Actually the answer is the germline spectrum, because it represents the historically prevailing bias throughout the human history. And so in this example that I showed you earlier, the reversal from this historically prevailing bias to this spectrum, this blue spectrum, is perfect, because the two types that were overrepresented became underrepresented, and all the four types, which were rare, became overrepresented. But in reality, we don't expect to see this perfect reversal, especially in a spectrum that has 96 types of mutations, like in the trimer level. And so this type can go up, but this can go down. So we have conflicting behaviors or conflicting changes in the frequencies as a contribution to the bias. And so we created what we call the bias reversal measure, which kind of summarizes the effects of all of these frequencies. It just sums how it changes and considers the direction into account, and it gives you one number. And this number, if it's positive, then we have a bias reversal, which by theory can give access to cancer driver mutations. But if it's negative, then there's a bias reinforcement, and that cannot contribute to that access. So here's the result on the 3 levels of analysis that we worked on: transition transvergence, 1-mer level, and 3-mer level. On the y-axis, you see the bias reversal measure, and these distributions are the distributions across different tissues. So again, yellow is normal, blue for non-hypermutators, and red for hypermutators. And the interesting thing is that on the 3 levels of analysis, we see a significantly positive bias reversal measure, and it's actually positive in all tissues, and that only applies in non-hypermutators. The distribution in all the other distributions are not significantly different from 0. So what we have so far is that non-hypermutators have similar spectra across tissue, like kind of a parallel kind of evolution of a spectrum, and they all reverse the bias. So that kind of meets the theory, but then hypermutators do not show that, which suggests that cancer driver mutations can be only accessed in this set simply because they have higher mutation rate. No other pattern is seen here. Okay, so we wanted to see what kind of mutational processes are underlying this reversal in the bias. And so because these spectra are quite similar, we pulled all the mutations from all the tissues together and performed the signature decomposition of that spectrum. And here I just want you to focus on the first column. I don't have time to discuss the hypermutators, decomposition for hypermutators, but I'm happy to discuss later. But here, every circle here represents one mutational signature from the cosmic signatures, and then that we found in the non-hypermutators spectrum. And the numbers here are just the names of the signatures. Each one represents one different signature. And the height of the center of this circle represents the bias reversal measure, as you see on the y-axis here. And the size of the circle is the weight of that signature, okay? So we see signature, so before saying about specific ones, you see that most of the signatures have positive bias reversal measures. So most of them are contributing in the reversal, okay? We see this signature 5 has almost half of the mutations, but it's simply one of the germline mutational processes, so it's not that interesting. But what's interesting is that 13, for instance, is a DNA mismatch repair defect related signature. And more importantly, and interestingly, 40. Signature 40 was recently attributed to hypoxia. But hypoxia only happens after the tumor has happened, right? So maybe it's kind of giving access, a second round access to cancer driver mutations or something. This needs more investigation, of course, but this is what we found so far. So to summarize what I've said so far, non-high permutator samples have similar spectra, and they all kind of reverse the bias, but no such pattern is seen in high permutators, which suggests that cancer driver mutations might have been accessed in non-high permutators due to the spectrum, the bias reversal in the spectrum, but that access happened in high permutators only because they have high mutation rates. So in the 1 minute I have left, I want to show you one more result, interesting result. So we also use the cancer genomics methods to detect positive selection in cancer genes, and that the goal was to see whether there are specific contexts that are more likely to be cancer driver mutations. And we did that separately for high permutators and for non-high permutators. And we found that 24 out of 96 contexts were significantly positively selected in non-high permutators, but in high permutators, only 2. This doesn't mean that there aren't driver mutations in high permutators, but the very high mutation rates can distort this signal anyway. But the interesting part is that we were able to detect these 24 contexts that are likely cancer driver mutations. And I want you to focus just on this negative correlation that we have here. The squares here represent the potential cancer driver mutations in the different contexts, the 24 ones. And the negative correlation here is between the germline spectrum and this axis measure, which represents how strongly positively selected this context is. Simply what this is saying is that the stronger a mutation is positively selected or leading to cancer, the less likely it is to happen in the germline, which suggests that cancer driver mutations might have been suppressed across human evolutionary history, but this definitely needs more investigation. And finally, I want to give you the QR code for our preprint in bioRxiv, and here is the list of funders, and thank you for listening.
Hello, everyone. Thanks for coming. My name is Israt, and I'm from the Queller-Strassman lab at WashU. And while some of you may have come to this talk with this title, but that was just to lure the computational biologists. But what I really am going to talk about, and I will use the techniques of genomic phylostratigraphy, but what I want to convince you today in this talk is how the question of evolution of form can be studied with developmental constraints. Before I begin, I would like to acknowledge my advisors, David Queller and Joan Strassman, my friend and frequent collaborator, Trey Scott, and for this project specifically, Dr. Barrera Redondo, who has developed the software for one of the phylostratigraphic methods that I'm using and has helped tremendously. I also want to thank all of you and many of you who have welcomed me and have had great conversations, so just imagine your picture there. But the question is, why am I in this session? Because I'm not really particularly going to be focusing on conflicts. And I think that has something to do with David Baum's spicy plenary, And as social evolution biologists, we probably have the most strenuous relationship with the concept of species. And not only are we in on this secret, we have some of our own. So where do we go from there? So species don't exist, organisms may not exist. But I hope to convince you by the end of this talk that there may be value to arranging your trains before you play with them, and how one can do that. So here I have taken the liberty of modifying this logo a bit. And I think a lot of the people here are here because they are interested in how organisms form. But at the same time, the things that are mentioned here, like genes, cells, and organisms, can also be grouped, let's say, into genomes, and cells are part of organisms. And organisms collectively, through shared ancestry, form species. But because I'm not going to offend two Davids in one talk, let's just say phylogenetically related group of organisms. And how I think they are united is by the concept of a body plan, which means basically comes from the German word of Bauplan, a blueprint, which means what part of your body is arranged in what particular way and when in development. So for example, insects have a head, a thorax, an abdomen, things like that. And it has played a central role in the evolution of development. And the good thing is, because these are such structural modules, a lot of the times they are encoded by the genome. So it's these patterns in time and space, in embryogenesis across all kinds of clades, have been studied and now are being studied in using different names like developmental patterning or transcriptome patterning and such. But the question of how organisms form and become into new organisms is older than the question of the origin of species. So von Baer, around 1930, gave this model where he said that the initial stages of an embryo are the most similar across all life. And as embryos grow, they become increasingly diverged. But unfortunately, Haeckel's, a thing that came out after the origin of species, confused a whole lot of people, created a whole lot of problems, but is not really the same as the progressive divergence that von Baer was talking about, which sometimes is called as the funnel model. So yeah, not that. And recently, developmental biologists have been studying, particularly in animals and sometimes in plants, this hourglass model where they say that as development progresses, embryos are very different at their initial stages, but some intermediate stage during development exists where they become increasingly similar, but from which they can diverge as they become finish embryogenesis or become adults. And then people have done inversed hourglasses, some people have done wiggly hourglasses, and it's quite complicated for my brain. But point is, people have figured out that there are some constrained modules in how genomes are organized, and they are being studied with sequence similarities. But not organisms develop in the same route. And while most of these studies have used this form of clonal development, where a single cell divides and then forms into the organism, there exists this charismatic microfauna called Dictyostelium discoideum, where individual cells could come together and then develop into a fruiting body, which has your germ cells as your spores and your somatic cells as your stalk. So I call it an organism. Before I begin, and that's the organism I'm going to be focusing the rest of this talk, but before I go to the system-specific questions, I would like to have a simpler model than all of the wiggly lines that I showed earlier. And what I would do here is that, let's say genes that are embedded in the genome, they get sort of founded over evolutionary time, but there is developmental time during which they are expressed in that particular organism. And what von Baer's model would say is that there are early constraints, and organisms, as they develop, become progressively divergent. So that would be this orange line. And the opposite can also happen, where genes that are really old, that have emerged in ancient lineages, they could be expressed much later in the development, whereas novel genes can keep appearing in the earlier stages of development, and that's what I'm calling the inverted funnel. Now, how does all these hourglasses stuff fit in here? I'm just saying, let's say, somewhere in the intermediate stages, there's either maximum convergence or constraints or maximum divergence. So for my purposes, we'll just focus on the early and the late stages. And I wouldn't be the first person to study this in Dictyostelium discoideum. In a previous research, Tian et al., they had found, by comparing transcriptomes from Dictyostelium discoideum and its relative Dictyostelium purpureum, that transcriptome seems to be converged later in development. So that would be something like this. As development progresses, transcriptomes become increasingly similar, and that would be sort of the inverse funnel model that I showed earlier. Where I come in here is I am approaching this question, but not only with the closely related species of Dictyostelium purpureum, but I am estimating the ages of all genes in Dicty's genome using the method called genomic phylostratigraphy, which is borrowed from the method of stratified rocks, where as you dig deeper, older lineages will appear and newer fossils are formed on top. So on the y-axis here, you would see that older genes are on the lower side, whereas as time progresses, younger genes are expressed later. And what I have found for Dictyostelium discoideum, so this is my phylomap, map, where depending on when the last homologue of a particular gene that is expressed in Dicty can be traced back to, that's the particular phylostrata that it belongs, and if something has emerged in all cellular organisms, that's older than the ones that can be only traced back to, let's say, Dictyosteliaceae. Now, when I combine these evolutionary ages to developmental ages of gene expression, using the method called transcriptome age index that Drost and Touts, those group have developed, you get something called a transcriptome age index. And here you will see that PS refers to the phylostrata that the gene belongs to, and the other is just a relative expression of the gene. So essentially what this means is if a transcriptome age index is lower, then the gene is older. If it's higher, then the gene is younger. And for Dictyostelium discoideum, I see a pattern like this. Here, the gray ribbon in the background is from 30,000 bootstraps, so you will believe me. And I see early constraints in Dictyostelium's development. And when I see this for each phylostrata, and so the gene's developmental ages when they are expressed, and this is for each hour, that's the x-axis, and the y-axis is your age indexes. You'll see that the newer genes that have emerged much later in development get expressed in later stages. But what does this mean for Tian's and my results? So when I have divided the phylostrata here in two categories. So if anything can be traced back to Dictyostelium, I have called that a younger gene. And if genes that can be traced back, that have emerged in Evosia or earlier, I'm calling those old genes. And you'll see that older genes tend to be expressed earlier, and new genes are expressed much later. Here, I wanted to map this to explicit developmental stages of Dictyostelium by taking a ratio of when this shift happens. So the black bars here refer to old genes, and the gray ones are genes that are younger. And I see that around the 6-7 hour point, that's when there is a huge drop in expression of older genes, and new genes, younger genes start to be expressed increasingly. And this shift is much stronger in the migration stages for Dictyostelium, which I had shown earlier. And eventually, in the culmination stage, the ancient genes are not expressed as much, and it's primarily taxonomically restricted genes that you see as development progresses. So in summary, genomic phylostratigraphy has shown that constraints in development may occur early in Dictyostelium, and genes get progressively diverged over time. Novel genes get expressed much later in development, and this shift can be mapped to key developmental stages. And there are a whole bunch of questions I am going to ask in the follow-up. I'm excited to talk about that if anybody wants to later on. And many thanks to my lab mates who have helped greatly over these years. And Margaret and Trinity, they are going to be speaking much more about the growth and asocial stages if you're interested in that in the predation session. Go check that out. Thank you.
So hi, my name is Kalyani Twyman. I am a PhD student at the University of St. Andrews with Andy Gardner, and today I'm going to be telling you about a project I've been working on. So one view of the history of life is that of a sequence of major transitions in evolution, and these were defined by Maynard Smith and Seth Murray as revolutions in the way that genetic information is stored and transmitted between generations, such that entities that were capable of independent replication before can only replicate as part of a larger whole afterwards. And within these major transitions in evolution are the major transitions in individuality, those that gave rise to a new form of individual at a higher level of biological organization. Examples of these include the evolution of genomes from single genes, the evolution of eukaryotic cells from separate unicells, the evolution of multicellularity, and the evolution of eusociality. And my work, if we zoom in, my work focuses on the evolution of multicellularity, and more specifically, the evolution of complex multicellularity. When I say the evolution of complex multicellularity, I mean multicellularity with division of labor, where cells specialize on certain tasks. So division of labor, and especially germ-soma specialization of cells on either reproduction or viability, is closely associated with the process of individuation. And a key characteristic of obligate multicellularity is that neither germline or somatic cells are able to survive or reproduce without each other. And interestingly, this division is also directly comparable to the division of labor that occurs in obligately used social groups, where individuals are split into specialized breeders or helpers. So one theory about the evolution of individuality and division of labor is Richard Michaud's group covariance effect. So he suggested that there is a consequence of group living and the life history trade-off between fecundity and viability that leads to the fitness of a group being augmented over the average fitness of its members. So his results suggest that group fitness, so given by this capital W, is the product of the average viability across the group big V and the average fecundity across the group big B. And this leads to the group's fitness being greater than the average fitness of its constituents, the little w, by an amount captured by this covariance term. And as there is a life history trade-off between viability and fecundity, the covariance term will be negative and thus represents a boost to the group's fitness. So Michaud argues that this is an emergent property of group living, this boost. However, it's a bit unclear how a group's fitness could be different from the aggregate of its constituent cells, and thus the effect remains conceptually obscure. So we wanted to investigate the same trade-off between viability and fecundity and its consequences for the evolution of division of labor to try to understand this covariance effect. So to do so, we modeled the transition to group living explicitly through considering two separate scenarios: a non-social group that does not share its viability and among its members, and a social group that does. So in this first scenario, the non-sharing scenario, we assume an individual's fitness arises from the investment in both viability and fecundity. And in order to separate the effort and the sort of the investment the cells are putting into and the resulting returns on fitness, we model this with the production of a viability enhancing and a fecundity enhancing good, where an individual's fitness is going to become the product of the two. And this becomes important a little bit later on. The group's fitness is then an average of all the group members' fitnesses. And here you see a covariance term as well. And this covariance term serves as a penalty for individual variation in a group, as it will be negative, as I said before, if there's any individual variation in the investment. So if we look at the other scenario, so the sharing scenario, the social scenario, if individuals do share their viability, we get these expressions for individual and group fitness, which are quite similar to the above expressions, but crucially, we see a lifting of that penalty for individual variation in a group given by that covariance term. And this loss of fitness penalty exactly recovers Michaud's covariance effect. And while he conceptualized it as this emergent property that can augment a group's fitness above the average of its constituents, we were able to find the same effect when we defined it precisely as that of the average. So a lot of what I just presented was contingent on this idea of viability sharing. But what is that? So we conceptualized it as the sharing of a viability-enhancing good with group members, which is very similar to the most common form of cooperation in microbes, the sharing of public goods. So just some examples of these microbial public goods are biofilms. So that's this picture here you can see, which allows microbes a way to capture nutrients from their environment. There's biosurfactants, which allows microbes to move, siderophores, which allows for iron scavenging, which consequently allows for some bacterial growth in low iron environments, and quorum sensing, which allows for communication. So our next question then became, if viability sharing is able to lift that inherent constraint to individual variation, does viability sharing evolve in Michaud's scenario? And if so, when? So we developed the model we used before, and the one that we derived the previous results for, and found this condition for the evolution of viability sharing in a group. And the left side of this inequality describes the benefit of ameliorating the group covariance fitness penalty, while the right-hand side describes the personal cost of an individual sharing, the one offset by the indirect fitness benefits given by relatedness R. And if we assume that the group covariance effect is going to lead to stabilizing selection in the ancestral trait as individual variation is being penalized by that penalty, the left-hand side of this inequality is going to tend towards 0. And that means the condition is only going to be satisfied when R is equal to 1 or in clonal groups. And this suggests something that we've called the clonality window for the evolution of viability sharing, a singularity where the conditions for it to evolve are met. And this just shows that visually, viability sharing is only evolving when groups are completely clonal. So just to go over what I've talked about till now, the group covariance effect is the lifting of an inherent fitness penalty for individual variation rather than an emergent property of it. And sharing viability is what can actually ameliorate this penalty. And we found that viability can be favored by natural selection when group mates are genetically identical, which we've called the clonality window. However, we were interested in division of labor. So now that we know when individual variation is not penalized, we wanted to know when division of labor itself might evolve, and thus complex multicellularity and perhaps individuality. So still within Michaud's scenario, we developed the model further to investigate the evolution of division of labor and how things like the group covariance effect and viability sharing might affect it. So first we incorporated these underlying functions for individuals' viability and fecundity. So here xij is going to be an individual's investment in viability, and 1 minus xij is then its investment in fecundity. K and kappa are just going to be scaling parameters, and the exponent a is going to describe the shape of the trade-off function, allowing us to capture potentially non-linear relationships between investment in fitness components and the fitness returns for division of labor. And to just explain that A a little bit more, an A between 0 or 1 or a concave trade-off is going to describe diminishing returns on investment or diminishing returns on specialization. An A of 1 describes a linear trade-off, and an A above 1 describes a convex trade-off or accelerating returns on investment. And then in order to capture evolution division of labor, we had to introduce one more underlying function to xij, where x is going to be the population average investment in viability. Epsilon is going to be +1 half the time and -1 half the time, randomly assigning individuals to specialize on either viability or fecundity. And delta is going to describe the magnitude of specialization on either of these, or then allowing us to capture the extent of division of labor. So after analysis, we find this to be the condition for when division of labor evolves. And again, here, A is the shape of the trade-off. The phi describes the ratio of investment in viability and fecundity, or x over 1 minus x, and alpha is going to describe the degree of viability sharing in a population. So it's hard to see right away what this condition is saying, but we can ask it a few questions. How does sharing going to affect the condition? And we find as the degree of viability sharing increases, the threshold value of A, or how accelerated the returns on specialization need to be for division of labor to evolve, decreases. So viability sharing is going to increase the likelihood of division of labor evolving. And then we can ask, at the extreme, what is going to happen when group mates actually fully share their viability among a group? And we find that the condition reduces to A is greater than 1, meaning that any accelerating returns to specialization lead to division of labor being favored when sharing is full. And this stems from that group covariance effect I talked about previously, because when viability sharing is full, we don't have that inherent fitness penalty, which is preventing the evolution of division of labor. So going back to that more general condition for division of labor, it relied on the balance of population investment in viability and fecundity. So that made us ask, what might this balance be optimally? So using the Taylor-Frank method of kin selection analysis, we found that the evolutionary stable strategy for individual investment in viability is given by this equation, where again, R is going to be relatedness and alpha is sharing. And this actually reproduces the dynamics of the tragedy of the commons. Individuals are going to be often favored to cheat and steal the viability from their group mates rather than creating their own. And this is going to happen when they're not clonal. And at the extreme of full sharing and no relatedness, the optimal investment in viability is going to reduce to 0, as everyone is favored to rely on other people's sharing. So if we take this optimal strategy and put it back into the condition for division of labor, we get this updated condition, where we now see that relatedness is going to modulate the evolution of division of labor in stable populations. However, it's only going to modulate it slightly, because at both r equals 0 and at r equals 1, it's going to reduce to this more simple expression, where A, again, is the shape of the trade-off, alpha is the level of sharing in a population. And in between these two extremes, the condition just becomes slightly more stringent in a sort of a U-shaped fashion. But what's most important here is that both of these conditions cannot be satisfied if alpha, or the extent of viability sharing, is equal to 0. Both expressions will end up with a value over 0 on the right-hand side, meaning they could not be satisfied by any A on the left-hand side. So if there's no sharing, the condition for division of labor cannot be satisfied, which means that it doesn't evolve in stable populations. And this means that the clonality window for viability sharing to evolve also serves as a prerequisite for the evolution of division of labor, which matches all the empirical evidence on the evolution of complex multicellularity, which is only seen to have arisen in multicellular taxa with clonal groups. So to update those takeaways we saw previously, we now find that the clonality window is also going to serve as a prerequisite for the evolution of division of labor in stable populations. And when sharing is complete, division of labor is favored with any benefit. And this case, therefore, represents the most likely scenario, might represent the most likely scenario for division of labor to evolve, clonal groups who fully share their viability. I just want to say thank you to my advisor, Andy Gardner, other members of the lab, as well as other people, funding sources, and then you guys for listening.
Well, thank you all for coming out, and thanks for having me here. These have been really exciting series of talks, and it's been exciting to hear everyone talk throughout this whole convention. My name's Joseph Dubie. I'm a postdoc at UT Austin, and I'm really excited to talk to you all about some really cool, simple experiments that I've been doing to explore how fusion, fission, and other bioenergetic homeostatic processes shape the potential fates of, in this case, selfish mitochondrial variants. And so we've talked a little bit about mitochondria, but this isn't a mitochondrial session, so I really want to make sure we're on the same grounds here. So what are mitochondria? First of all, they're an ancient endosymbiont. They've been evolving with us for over 1 billion years at this point. They are our potential first friend. But like all good friends, they keep a certain degree of separation. They still have their own genomes despite all this time, right? They're small, greatly reduced. In the case of our study, Organism, they're only about 13 kb. And they're circular, and they still replicate independent of the cell cycle, right? So they're continuously replicating. You're getting multiple rounds of replication, both of the mitochondria and the mitochondrial genomes. And there's a lot of them, right? In an individual metazoan, you can have hundreds, well, singles to hundreds of mitochondria. Each of those will have singles to multiple mitochondrial genomes. And so you can start to see where, this is a conflict session. You can start to see where some of these conflicts might start to arise. But keeping the story nice and friendly, continuing, as we all know, mitochondria, powerhouse of the cell. For the purpose of this talk, we're going to assume that their main role is energy production. But we also know that energy production and energy need is not consistent throughout an individual cell, and it's not consistent throughout the life of that cell. And so mitochondria are constantly fusing, dividing, and replicating in order to help the cell meet those energy requirements. So having set up all of this, what is the source of our conflict? And the source of this is the fact that there are kind of 3 different players in this situation, all which have subtly different, or in the case of the mitochondria, drastically different selective pressures. On an organismal level, we want to have, the most productivity, the most of our progeny reaching the next generation, and we want to basically not have to be too stressed throughout our life. The nuclear genome seems to work pretty concurrently with that most of the time. But when we are talking about the nuclear genome and the selective pressures on it with regard to the mitochondrial genome, it wants the mitochondrial genome to be efficient, under its control, and not mutating too rapidly. To contrast that, from the perspective of mitochondrial selection between mitochondrial genomes within a cell, they're going to be more fit if they're replicating faster, preferentially getting into the next generation, and finally, not easily detected by their host. And that's because if there's like a deleterious variant that's present, if it's able to hide from the host cell's kind of cleanup processes, it's going to be able to kind of persist despite harming its host. So if this conflict exists, why is it that we don't just have mitochondria constantly just melting down within every single cellular generation? And while experimental evidence in this is still ongoing, people have been thinking about this problem for a very long time. And so I'm going to talk about some of the different levers that a host cell has to modulate this selfishness within an individual mitochondria. So the first thing it can do is if you have deleterious mitochondrial variants and they're not meeting your energy needs, you just produce more, right? That's the simplest answer. However, this is going to favor those selfish variants. If you have a variant that's poorly performing and you produce more, now you need even more, and it just kind of like cycles up until eventually that selfish variant takes over the cell. To counter this, we have selective mitophagy. The host cell can target dysfunctional mitochondria, especially on the basis of breakdowns of membrane potential, and destroy those ones so that they can basically remove them from the system. And this is very tied up in the process of biogenesis, and so it kickstarts the production of more mitochondria. However, this is very, very dependent on the cell's ability to detect these deleterious mitochondria. And so the other lever that it has that I'm going to talk about during this talk is membrane reorganization, right? And so what it can do is it can either fuse mitochondria, creating more efficient, more networked mitochondria. This is going to increase homeostasis within the cell, make it healthier, but it's going to obscure those selfish variants. Now they're co-occurring within individual mitochondria with healthy, good productive mitochondrial genomes, and mitophagy is going to be less efficient at selecting against those. Alternatively, we can have mitochondrial fission. this is going to expose individual mitotypes to this selective mitophagy. However, it's also going to lower overall fitness because now we have mitochondria that are poorly performing. And so without increasing the healthy mitochondria, we're just going to see dysfunction. And with them being less networked, they're also less efficient. So those are kind of the ideas that have been out there. There's a lot of people have been there's a lot of really good theoretical work and some experimental work. But I wanted to really directly test these ideas. And so there's 3 questions we're going to be going through throughout these slides. How does disrupting fission affect selection against a selfish mitotype? You can see the prediction is that it'll increase it. Disrupting fission, we expect it to do the opposite, decrease selection against the selfish mitotype. And how does, in this case, we're going to increase mitophagy to see if that can help to select against the selfish mitotype. All right, so then how are we going to do that practically? Well, first we're going to go ahead and create heteroplasmic lines, so they're going to have a selfish variant and a healthy wild-type variant present within the same cell. Then we're going to go ahead and disrupt these homeostatic processes. And finally, we're going to measure the effects that these disruptions have on the intra-individual dynamics of this mitotype. So here's our study system. This is UADF5. I'm going to also refer to it as delta mite often interchangeably throughout this entire talk. And that is because it has this big deletion that you can see on the right here. It's about 3.5 kb, so it's pretty close to about a quarter of the mitochondrial genome is just deleted, right? So that's 4 genes and a handful of tRNAs. They're just completely gone from these. They persist as heteroplasmies. As you could imagine, missing a quarter of your genes from your mitochondrial genome does not lead to fixation. It leads to a bunch of dead worms. But it's selfish enough that it's lab stable. So you can keep populations of these worms in benign conditions, and the heteroplasmic frequency is going to stay right around 55%, which means that this is a great system for testing these questions. So, like I said, we wanted to create these lines with the disrupted homeostasis. And so C. elegans is a great system. It is a facultatively outcrossing hermaphrodite. And so what we just do is we take a hermaphroditic worm that contains this selfish variant, and we outcross it to male worms, which contain each of our variants of interest that we'll go into in just a second, thereby replacing the nuclear genome with one that has these homeostasis processes knocked out, and pushing through a mitochondrial genome that has our selfish variants present as a heteroplasmy. We then did a bunch of fitness assays. Most of those are very intuitive. They'll be ones that you will understand when I say what they are. But in particular, I wanted to point out this respiration assay because one of the things we measure in these lines is going to be mitochondrial respiration. And the way that we do that is by putting 200 worms into solution, putting that into this machine called an O2K respirometer, allowing them to acclimate. This kind of measures basal respiration within the solution. We then add sodium azide, which is a complex 4 inhibitor. This is going to shut down all of our mitochondrial respiration. And then the difference between those two states is going to give us our mitochondrial respiration. This is telling us how much of the respiration that's going on is due to the mitochondria itself. And then finally, the really cool part is this intra-individual competition. We propagated 25 replicates of each of these different strains that we created for 5 generations, and we used DP in a bottleneck population, so a single hermaphrodite every generation. And we used dPCR to go ahead and measure heteroplasmic frequency of our variant of interest, this UADF5. And we kind of have 3 different predictions for what might happen over that time frame. A neutral prediction where the heteroplasmic frequency doesn't really change, selection against the heteroplasmic frequency when it goes down, and selection for where it goes up. So the first variant that we took a look at was this FZ01. It is a reasonably sized deletion of that gene. This is going to reduce outer membrane fusion, creating a greater number of small punctate mitochondria, which means that they're going to have greater chance of having just one mitochondrial variant present within each organelle. And this should increase sensitivity to UADF5. Kind of going a little faster here, but when we look at the fitness of this, this is normalized productivity, so normalized to a wild type average. And you can see that all of our lines are the FZO, the double mutant, and then the heteroplasmy by itself. And you can see that the FZO mutation is actually more deleterious than the variant that we're interested in. But when it is together with the selfish variant, it actually has slightly increased productivity. You might think that's compensatory, but then when we look at time to adulthood, you see that there's actually no difference between all of our lines, and so it's not creating greater fitness there. And when you look at respiration, it actually has the exact opposite mitochondrial stress phenotype here, so really low respiration in the UADF5 means that we're not producing a lot of energy in these lines. And then really, really high means that it's kind of always acting in a stressed state, right? So it's not really going to be able to respond to any kind of stresses because it's already working near its capacity. So when we do that competition experiment that I had described, what we see is like a very, very clean result. We're going from about that 55% that we started at that I was discussing, and then over those 5 generations, we're down to about 5% within individual worms. This is a very clear sign that disrupting this membrane fusion is also selecting against the heteroplasmy, right? So let's go ahead and do the opposite, right? Now we're going to go ahead and give ourselves a more networked mitochondria. This is being done by disrupting DRP1. Once again, a deletion. Going to reduce mitochondrial fission, creating more networked mitochondria. These should be more efficient, but they should also mask the phenotype of this UADF5 within the lines. When we look at, sorry about that, productivity here, once again, it actually kind of looks like the double mutant is like a very fit line here, right? If you're just looking at productivity. However, when you look at time to adulthood, you can see these are taking almost twice as long to reach adulthood, right? So a good reminder, measure multiple phenotypes. Don't just bank on one. And when we look at mitochondrial respiration, it's not any different. And this is just the single mutant. This is not the double mutant. It's basically the same as our mitochondrial deficient lines. And just like we predicted, when you compete these worms, you see the heteroplasmic frequency go up. There is something interesting about this, and I will note, after backcrossing, it was down all the way at 19% on average. So there was something about the backcrossing process that reduced the heteroplasmic frequency. But then when they were just propagated on plates in a bottleneck population, the frequency went right back up. And finally, I'm going to talk about this DCT1 mutation. This is actually increasing the amount of mitophagy that's happening within these lines. Then it does this by modifying the area just upstream of the genome to kind of like increase the expression of this DCT1. It should increase mitochondrial turnover, and therefore more mitophagy. We should see lower levels of UADF5. It's actually, I have a mutation on my slide. This is not the right graph. But just imagine a graph that has 3 box plots that are basically identical to one another, because we don't see like any effect of fitness of this mutation. And even when we look at respiration, we can see that the DCT1 lines are somewhat intermediate between our UADF5 line and our wild type line, but not distinct from either one. So it's sort of healthy, sort of. And when we look at these DCT1 deletions and at this competition, what we see is like right away you'd look at this and say, okay, looks neutral, looks like nothing's happening. But once again, point out, during backcrossing, that frequency went way, way down, all the way down to about 10%. And it stayed there throughout the whole competition. And the interesting thing is that we went back to try and select for this higher frequency, trying to get a more intermediate one to give it a much better shot, right? But every single time when we got to F2 homozygous worms, the frequency was right back down to that 10%. And so it seems like there's something about being a homozygous for this that's really, really strong selection against this mitotype. And so here's all of my stuff. I think that I'm basically over time at this point. But just reiteration of everything, modulating all of this had very strong effects on the fate of these mutations. And so this is like really, really important for the overall fate of any particular mitotype, and that's kind of where we're going to be going next with this. Thank you all. Thank you to all of my lab members, most of which are here. And if you would like to ask me any questions, here's my email. Please hit me up. All right, thank you very much.
I'm Dinah Davison, and I'm going to be talking to you today about plastic changes in cooperation and how they may shape the evolution of multicellularity. The evolution of multicellularity is a major transition in evolution. During this transition, cells come together to form groups. These groups evolve increasing levels of integration, and cellular specialization evolves. Now, during this transition, selection transitions from acting at the level of the single cell to acting on the level of that multicellular group. And cooperation, conflict, and conflict mediation drive this change. So cells come together in these groups where they're cooperating. This sets the stage for conflict, and conflict mediation mechanisms then evolve that stabilize that group. And an important example of a conflict mediation mechanism that's key to the evolution of multicellularity is that of cellular specialization. Specifically, if you think about somatic cells, they're specialized on the survival of that organism as a whole, and they don't reproduce. So if you have specialized somatic cells and specialized reproductive cells, the fitness of any given cell is dependent on the fitness of that multicellular organism as a whole. So we know that mechanisms that are shaping the evolution of multicellularity change over evolutionary time. We see this with the evolution of cellular differentiation. But can mechanisms that mediate cooperation and conflict among cells also change on shorter timescales? Can you see dynamic changes in cooperation and conflict among cells in different key steps in the evolution of multicellularity, specifically during the evolution of group formation and the evolution of cellular differentiation? Moreover, could such plastic changes be intermediate stages in the evolution of multicellularity? So to investigate these questions, I use the Volvocene green algae as a model system. They're a great model system for studying the evolution of multicellularity because this clade contains unicellular species like Chlamydomonas, undifferentiated multicellular species like Gonium or Eutorina, and species with multiple different types of cells, like you see with Pleidorina and Volvox, which have specialized somatic cells, which are those really small dots that you see in those organisms. So let's start by thinking about the evolution of group formation. Now, in the Volvocene algae, you can see this phylogeny of the algae on the right. You don't need to worry about those specific names. But we know that multicellular developments has evolved at least twice. And you see that with the repeated origin of multicellularity, those purple nodes there. But that's the evolution of obligate multicellular development. Group formation in unicellular green algae is actually ancestral to the Volvocene algae. Numerous green algae species that are typically unicellular will form these groups in response to environmental stress. And we see that in Chlamydomonas, which is that unicellular species that's an outgroup to the rest of the Volvocene green algae. And Chlamydomonas forms groups via two different pathways. It can form groups via aggregation, so cells will come together to form this aggregates, similar to how dictyostelium cells can come together to form a multicellular organism. And this sets the stage for conflict, because these cells aren't necessarily closely related to each other. And here you can see an image of a Chlamydomonas aggregates, and you can see on that graph there how Chlamydomonas cells come together to form groups right after a predator is added to the culture. So that's that light green line. Then those cells actually come apart at night when they're dividing, and then they aggregate back together during the day. Chlamydomonas can also form cell groups via clonal development, so that's cells staying together after division. Those cells are all going to be very closely related to each other because they're all descending from that same unicellular bottleneck that they develop through. And this sets the stage for increased cooperation because of that high relatedness among cells. And here you can see some images of Chlamydomonas clonally developing groups that have developed in response to salt stress. So we've got 3 different developmental pathways that Chlamydomonas can travel down. Most of the time, any given single cell is going to divide and give rise to more single cells. So that's the most common pathway right in the middle there. Chlamydomonas can also develop along this pathway of clonal development or along this pathway of aggregative development. So how can we use this to understand the evolution of group formation? Well, we know that Chlamydomonas cells form groups in response to harsh environments. We also know that there's a species called gonium that's typically multicellular, but gonium can become unicellular in response to harsh environments. Other multicellular volvocine green algae species are always multicellular regardless of the environment. So what we appear to have going on is, right, you have an ancestral state where there's multiple different developmental pathways you can travel along. One of them is group formation that sets the stage for more cooperation. One is group formation that sets the stage for more conflict. And we're seeing the loss of that developmental pathway that sets the stage for increasing levels of conflict. And we're still working out the exact pathway by which you go from may have gone from a Chlamydomonas-like ancestor to something that develops multicellularity regardless of the environment. But we do see that these developmental pathways involving increasing cooperation among cells may have been maintained and stabilized. So next, we're going to talk about the evolution of cellular specialization. So in the volvocine algae, specialized somatic cells have evolved multiple times. You can see that on the phylogeny everywhere that you have yellow and purple nodes. And if we look just within this one small subclade called the EVP clade, we see the repeated evolution of somatic cells. And within this clade, you've got 3 polyphyletic genera. You've got Uterina, which are characterized as being undifferentiated, and you've got soma-differentiated Platorina and Volvox, which have these small little somatic cells that don't reproduce. So I'm focusing on a species of Uterina in order to examine whether or not you can see plastic changes in cooperation and conflict mediation in the form of cellular specialization. So I took Uterina elegans and I subjected it to 2 hours of cold shock. And I found that these cold shock colonies will develop somatic cells. Additionally, some of the offspring of those cold shock colonies also develop somatic cells. However, the grand offspring of the cold shock colonies are indistinguishable from controls. So this differentiated phenotype is plastic, it's not stably inherited, and you've got this role of maternal effects occurring here. So I just showed you that Uterina can develop these plastic somatic cells in response to the environment. So we're seeing this facultative increase in cooperation among cells. But could this plastic response be an intermediate stage in the evolution of differentiated multicellularity? Could the plastic response be stabilized? And that's how you get obligate cellular differentiation. Well, to examine this, I used experimental evolution. So I established multiple Uterina lines from a single colony, and I cold shocked them every other generation and transferred them to fresh media. And I repeated this 5 times, put them back in culture collection, did the same thing, cold shocked them every other generation, repeated this 7 times, put them back in culture collection, and 5 months later, which corresponds to somewhere between 40 and 75 generations, I grew these lines in flasks and I imaged them. So all the data you're going to see comes from colonies that haven't been exposed to that selective stressor for dozens of generations. And I found that somatic cell regulation evolved following those instances of repeated cold stress. So if you look at the control lines on the bar on your left, you see that the majority of colonies are undifferentiated, that's shown in dark green, although there is a small proportion of uterinas that have somatic cells at any given time. Compare that to the lines that underwent repeated cold stress, where you see that a much larger proportion of colonies have somatic cells. And when I broke this up to look at individual lines, I saw that same pattern consistently repeated across my lines. Next, I pulled out individual colonies from these lines, and I found that in one of the lines derived from one of these subclones, somatic cells were constitutively expressed. So almost all of the adult colonies I examined in this line had somatic cells, and that continues to be true over a year since the last time they experienced cold shock. So this shows that plastic cellular differentiation can be stabilized, and it can be stabilized rapidly. So how does this help us understand this transition from facultative cooperation conflict mediation to obligate conflict mediation in the form of the evolution of cellular specialization? So let's think about the development of uterina. So I've circled a single cell there in light blue, and we're going to track its development. So it divides, it undergoes inversion. You have a daughter colony developing, which hatches out. It grows and develops into an adult colony. We can simplify that developmental process with this diagram here, which is similar to the small diagrams I showed you earlier, where we're starting from a single cell, that's that green circle. Most of the time, it travels along the developmental pathway represented by that thick arrow to give rise to an undifferentiated colony, although occasionally you get the development of small somatic cells. And we can further represent this as a ball rolling down a hill in this simplified version of Waddington's epigenetic landscape. Now compare this normal uterina development to development after cold shock. So the addition of cold shock changes the shape of that developmental landscape so that now differentiated colonies are much more likely to develop. Now, during my experimental evolution study, I stabilized that previously environmentally induced developmental landscape so that you see the consistent development of both differentiated and differentiated colonies. And in line 11A5, the one that's obligately differentiated, you see further changes in that developmental landscape so that you consistently get the development of somatic cells, which is very similar to the developmental pathways that obligately differentiated species like Pleiadorina might develop along. So what I've showed you today is that dynamic plastic changes in cooperation among cells may have shaped the evolution of multicellularity. And this is true for that key step of group formation as well as in that step of cellular differentiation. And I also showed you that these plastic responses can be stabilized such that increased cooperation is consistently present. And I showed you that in this example of the experimental evolution of cellular differentiation. So this work taken together indicates that increased cooperation in response to the environment may be key intermediate stages in the evolution of differentiated multicellularity. So I'm currently in the middle of examining gene expression in plastic and obligate somatic cells in multiple Volvocene algae species. And I'm preparing to modify my experimental evolution protocol to collect additional data to understand the pathway by which obligate somatic cells evolve. So I'd like to thank a number of collaborators, my PhD supervisor, Rick Michaud, my postdoc supervisor, Brad Olson, thank Aurora Nedelku, number of other people who've worked on this project, my funding sources, especially the NSF, and all of you for being here today. And I'll take any questions you may have. Thank you.
Hello. Thanks for being here. I'm Tatiana, and I'm going to present to you a story about genetic conflicts in bacteria. So I've recently moved to St Andrews in Scotland, but most of this story has been developed over a few years as a side project during my postdoc in Exeter. And I did this in collaboration with Andy, who is here, and Angus Berkling. Oh. So I study conjugative plasmids in bacteria. And plasmids are DNA molecules which replicate independently from the chromosome. And they can transmit to daughter cells at cell division. And vertical transmission already can lead to some conflicts, genetic conflicts at transmission. But the plasmids I study are conjugative plasmids, which additionally are able to transmit horizontally. This doesn't do the same. I don't see this.
You see the slides?
It doesn't move the same way on this compared to here. I've not managed to put it on presenter view, but sorry.
Yes, so this is one slide. This is the next slide.
Oh, this is the
I think.
presenter view. Okay, sorry.
Or
Yeah, sorry, thanks. So conjugative plasmids encode a conjugation machinery which allows them to transfer horizontally from donor to recipient cell after cell-cell contact. This has a cost to the host, so it is in itself a parasitic behavior from plasmids. And additionally, those plasmids can carry cargo genes or accessory genes, which typically confer to the host benefits in specific environmental conditions. And the main example of this is antibiotic resistance, which confers resistance to antibiotic treatment. And so this makes plasmids mutualist in specific environmental conditions. So I'm interested specifically in the evolution of horizontal transmission itself, which will be central in the genetic conflicts between plasmids and chromosomes. And plasmid transmission rates are strikingly variable across plasmids. So this was a meta-analysis that compared conjugation rates across plasmids and across bacterial background. And they showed a striking diversity of spanning 14 orders of magnitude between plasmid transmission rates. So one question we have is, in which conditions will plasmids evolve to transfer basically all the time for the high ones, or almost never? And one main hypothesis about this has been around in the literature for a while, and is that it's beneficial for a plasmid to invade in costly horizontal transmission, basically when horizontal transmission will allow it to invade new hosts, so when new, naive, potential new hosts are often available in the environment. So we decided to test this experimentally using an experimental evolution approach. And to do this, we manipulated the opportunity for horizontal transmission for plasmids. On the bottom line, you see the control so pure vertical transmission control, in which we passage plasmid carrying cells over and over again, and there is never opportunity for horizontal transmission. And then we added a few different increasing horizontal transmission treatments, in which every day we added an increasing proportion, up to 99% of plasmid-free cells to the population. And we had no antibiotic selection during evolution, so plasmid-carrying cells could go extinct, and plasmids had to transfer if they wanted to be maintained. I did this with a model antibiotic resistance plasmid, R1 in E. coli, and this plasmid has an antibiotic resistant gene, ampicillin resistance, which is also the way that we use to identify plasmid-carrying cells by plating on antibiotic medium. So first, this is the population dynamics that we observed during evolution. So in the absence of immigration, so in the lightest thread treatment on the top, we saw stable plasmid carriage, so vertical transmission is efficient. But with increasing immigration, we observed a steeper and steeper decline in antibiotic resistance over time, with total loss of antibiotic resistance at the end for the highest immigration treatments. So then we asked have plasmids evolved their transmission rate, and does it depend on treatment during evolution? So to answer this question, we selected one single plasmid carrying clone at an intermediate time point in each of the replicate lineages. and characterized their phenotypes and genotypes. So first, I measured plasmid transmission rate by doing simple conjugation assays, where you simply mix plasmids carrying donors and recipients for a few hours, and then you plate them. And you measure transfer rate as the amount of transconjugants, so recipients that received the plasmid, divided by both donors and recipient densities. And this is the results we observed. So the black line is the ancestor conjugation rate, and each clone is ordered by immigration treatment with high immigration on the left, and the y-axis is a log scale. So according to the predictions of the theory, what we observed is that specifically in high immigration treatments, plasmid evolves towards higher conjugation rates. And this does not happen if plasmids evolve with only vertical transmission. So we were happy about this phenotypic data, and then we sent those clones to sequencing to identify genetic mechanisms.
Okay.
Thank you. I'm so sorry. I think I didn't put the other. So the main genetic mechanism we observed was not what we expected, which would have been specific mutations in transfer genes, but instead it was mutations that affect plasmid replication overall, and those are mutations that increase plasmid copy number. And the way that we think this works is that plasmid transfer needs basically expression of the transfer machinery on one side, and then this transfer machinery to recognize plasmid DNA at a specific small sequence, the origin of transfer, and those lead to transfer. So with increased gene dosage and plasmid copy number, there is both increased gene dosage of the transfer machinery and also more DNA to be available to transfer, and those combine to increase plasmid transfer rate by roughly 20-fold in total. So then why do we care about this in terms of evolution? So one key point of this mechanism of evolution is that it will affect all genes on the plasmids and not only the transfer genes. And in particular, it will increase expression of antibiotic-resistant genes. And this is well known to be able to increase antibiotic resistance. And indeed, when we plate whole populations evolved in our different treatments on high levels of antibiotics, so at concentrations at which the ancestral plasmid does not provide resistance, we observed that specifically the populations that evolved in the presence of selection for transfer now have a large proportion of cells that can grow at very high levels of antibiotics. So the conclusion at this stage is that when cells evolve in the total absence of antibiotics, but in conditions that promote horizontal transmission, we see the evolution of increased horizontal transmission, which is costly for the host, but at the same time, because of the pleiotropic effects of the mechanism, we have high level of resistance to antibiotics, which is beneficial for the host in some conditions, but also quite scary in terms of antibiotic resistance management. So I'm now, during the time I have left, going to show that actually, so this is what we published in 2021, but we missed a whole other layer of genetic conflict happening within those populations, which turns the story on its head. So first, I don't have time, but so the population dynamics data were slightly confusing. So the collapse in antibiotic resistance actually only happened after 12 days. It was pretty stable before that, which was weird. And the other data we had was detailed results from the genome sequencing data. So this is results of Illumina sequencing, where we map the reads from plasmids across the sequence of the plasmid. So the black line is again the ancestor plasmid, and most of the evolved red lines, red plasmids, have increased coverage overall, corresponding to increased plasmid copy number. However, there is a decrease that you can see in some of the region of the plasmid. And this region is the antibiotic resistance region. So we took a lot of time to figure out what was happening here, because we were thinking in terms of bacterial clones we sent for sequencing and couldn't think of variability within clones. But actually, because we now have high copy number mutants, we get coexistence of full-length antibiotic resistance plasmids and deleted high copy number antibiotic-sensitive plasmids within cells. So those cells are antibiotic resistant, but deleted cells with just the deleted plasmids are antibiotic sensitive, meaning we cannot trust anymore antibiotic resistance phenotypes to tell us if there is a plasmid in the cell. And so indeed, when we go back to the evolved populations and when we characterized these populations, the endpoint ones, in the absence of antibiotics by PCR to search for plasmid reads in individual colonies, we found that those populations are actually all full of deleted plasmids. So what we thought was plasmids not being able to transfer fast enough to compensate for immigration was actually an invasion of deletion variants which displaced the antibiotic resistance plasmids. And then I did a few competition experiments to understand where the benefit comes from for those deleted plasmids compared to the wild-type ones. Is it horizontal or vertical transmission, or both? So I won't get into details on the experiment itself, but basically, so this is an experiment looking at vertical transmission. And what we saw is that the wild-type plasmid is stable on its own, and is also stable in competition with a deleted plasmid if the deleted plasmid has a low copy number. But its antibiotic resistance declines quite fast if the deleted plasmid is at high copy number. So high copy number provides a benefit to deleted plasmids for vertical transmission. And then we did a similar experiment, but now in a setting where transmission will happen mostly by horizontal transmission to naive recipient cells. And here we saw that the antibiotic resistant plasmid on its own invades a population in a few days, in 4 days. But as soon as we also introduce deleted plasmid in the population from independent cells, this is totally stopped. And when we zoom in at an intermediate time point, we find that the wild-type plasmid hasn't spread yet really in most of the population, but all of the deleted plasmids have. And this also includes deleted plasmids which are not high copy number. So this suggests that the mechanism for increased horizontal transmission is here just due to the fact that those plasmids are smaller and transmit faster. So to conclude, what we saw here in the end was that conditions that promote horizontal transmission do lead to high transfer rate, highly transferable plasmids. And because of the mechanisms for this leads to both high-level resistance to antibiotics in a small proportion of the plasmids, but actually at the population level, mostly to a deletion of resistant genes and displacement of resistance by deleted plasmids. And at a more general level, this means that we can sort of see those deleted plasmids as a defense system against the other wild-type antibiotic resistance plasmids, which fits with what has been observed recently in terms of defense systems where people look at defense against mobile elements in bacteria and realize that actually a lot of those defense systems are competing other mobile elements within cells. Thank you.
Hi everyone, my name's Anne-Lise, and today I'll be talking about Tasmanian devils and transmissible cancer. But first, I wanted to ask you a question. Can you remember the last time you had to share a house with flatmates? I'm sure you've all had a similar experience where on occasion you would have this one housemate who forgets to put the bins outside or who leaves the dishes completely dirty in the sink. Well, imagine cancer as being that, but 1,000 times worse. So now your flatmate is inviting other random people around. They're all eating your food, and one day you come home and your home is literally overtaken by all these obnoxious people that you have no idea about who they are. Well, this is a very clever analogy that I didn't come up with, but Athena Actibis came up with it in her book, The Cheating Cell. And it's a very clever analogy for cancer. So you can imagine cancer as being a loss of cooperation among cells within an organism. And this is due to cells having a tendency to increase their own fitness at the expense of the organismal fitness. And obviously, in that case, we find ourselves in a within-organism conflict. So you might ask me, why do we care? Most of the time, cancer dies with its host, so what's the problem here? Well, here comes my model species and system, which is the Tasmanian devil. And these lovely guys are marsupials endemic to Tasmania and Australia. And they've got a pretty big problem. They've got a rare form of transmissible cancer that's named devil facial tumor disease. And you can pretty much imagine why it's named like that. So it is characterized by these big tumors that grow on the face of devils. And the cancer itself is a direct transmission of cancer cells between individuals, and that happens when they bite each other through social interactions. So because one cancer would have already been like a simple problem to solve, these guys actually face two completely independent transmissible cancers that are genetically completely distinct, which from now on I will name DFT1 and DFT2. So the first cancer arose around 40 years ago in north of Tasmania and is now found virtually everywhere in the island, whilst the second one is still relatively limited to this peninsula south of the island. So the problem we're having is that this first cancer has caused significant population declines, and we're not quite sure whether the second one would be as dangerous as the first. And on top of that, within the area where DFT2 is present, there have been cases of DFT1 and DFT2 co-infections within a same devil that have been recorded. So, which brings me to the aims of my PhD. We expect competition to occur between those two cancers, at least for hosts and within hosts. And that brings me to my three aims, which are, apologies, I'm not being able to see what I have. Yeah, now it works. Okay, sorry for that. So my three aims, which are to characterize the in vitro growth kinetics of these cancers, and also to characterize the metabolic pathways that drive the growth of each cancer, and finally looking a bit more into detail at the gene expression behind those pathways. So I'm facing the same problem as the previous speaker. It's okay, I'll deal with it, sorry. Okay, so which brings me to my first experiment where we grew DFT1 and DFT2 cells in vitro. We did that in monocultures, and we counted the cells each day for approximately 2 weeks. And then we fitted a logistic model in order to estimate two parameters, the first one being growth rate, so how fast the cells grow, and the second one being carrying capacity, or how dense the cells can get in culture. And then we also performed co-cultures. So by labeling one of the DFTs with GFP, we were able to culture them together and to count them respectively and see how they would perform in competition. We fitted a Lotka-Volterra competition model in order to estimate competition coefficients. And this brings me to my first set of results. So in monocultures, you can see in red DFT2 and in blue DFT1. And you can see that the growth rate for DFT2 is approximately twice as high and significantly higher than for DFT1. So we know that DFT2 grows faster. And then when looking at carrying capacity, we can see that DFT1 is able to reach higher cell densities than DFT2. So DFT1 grows bigger. So now you might ask, what happens when we smash both cell lines together in co-culture? Well, DFT2 is able to take over, and we see quite a bit of variation in these results because we did try different starting ratios of cells. But in red, you can see alpha, which is the parameter characterizing the competitive effect of DFT2 on DFT1. And then in blue, you can see the competitive effect of DFT1 on DFT2. And obviously, the red dot is higher up, and it's a significant difference between both, indicating that DFT2 is a better competitor. All right, so what might be driving this competition? So I'll go back a bit to basic biochemistry here, but for cells, there's basically two ways to produce energy. There's glycolysis, which results in energy in the form of ATP, and then there's also when you completely oxidize the glucose and run the electron transport chain, that also leads to ATP production. So normal cells, they do both of them. But cancer cells are very well known to upregulate glycolysis in order to produce ATP at a much faster pace. So what we did is inhibit glycolysis and see how the cells would cope. Then we also quantified lactate, which is a byproduct of glycolysis. And at last, we selectively inhibited either the electron transport chain or glycolysis in order to understand where most of the energy from the cells comes from. So when it comes to results, this is what we observed when inhibiting glycolysis. We observed significant mortality in both DFT1 and DFT2, indicating that glycolysis is indeed important for these cells. Then when quantifying lactate production, we did see a slight but significant increase in lactate production in DFT2 cell lines, still represented in red here. And indicating that DFT2 produces more lactate, which might be due to a higher rate of glycolysis. And then when looking at total energy production in the form of ATP, we did see that DFT2 produces more ATP than DFT1. And then in order to understand where that ATP comes from, we selectively inhibited those two important pathways, which are glycolysis and the electron transport chain. And we can observe that for DFT2. Inhibiting glycolysis leads to a significant decrease in the amount of ATP produced, but not when we inhibit the electron transport chain, indicating that DFT2 probably relies more on glycolysis than on the electron transport chain. When it comes to DFT1, we saw a significant reduction in ATP production in both cases, so probably indicating that in some way DFT1 is still relying on oxidative phosphorylation, but not as much as DFT2. All right. So now we wanted to ask, where does this change in glycolysis rate come from in DFT2, and what is genetically driving that? So we did some transcriptomics, and I'm not going to show you all the results of this for the sake of time, but what we did was RNA sequencing and compared directly DFT2 to DFT1 cells, and we also then had a look at biopsies in order to have some in vivo data and trying to understand whether this is actually what's happening in the devils. So here's a bunch of glycolysis-related genes that are differentially expressed, but the one I want to focus on now is called PFKM, which is the phosphofructokinase mutase. This is a gene that is responsible for converting fructose 1,6-phosphate into, fructose 6-phosphate into fructose 1,6-biphosphate. And this is very important as this is a gene that is this enzyme, is responsible for what is known as the committing step of glycolysis. So once this enzyme enters into action, all the fructose 1,6-biphosphate it's producing will be turned into pyruvate ultimately. And I went a bit quick and forgot to say that this was the most differentially expressed gene between DFT1 and DFT2 when it comes to glycolysis, being upregulated in DFT2, and we did observe the same thing in vitro as well, in vivo as well, sorry. All right, which brings us to my summary. We found that DFT2 does grow faster than DFT1, and that this is likely driven by a higher rate of glycolysis, possibly itself being driven by this upregulation of PFKM. This in turn leads to an increase in lactate production, but also in energy, so probably explaining how DFT2 is able to gather all the energy and building blocks it requires in order to divide faster than DFT1. Lactate production in itself is quite interesting as well, because it will acidify the tumor microenvironment and could have big implications for immune evasion, for instance. So all these results are quite interesting because we did, previous research noticed that DFT2 tumors did tend to occur more, so although most of the tumors occur in the face of devils, the DFT2 tumors tend to occur more often on the body of devils than the DFT1 tumors. And this could be interesting as it might able DFT2 to find a different niche since it emerged in a landscape where DFT1 was already mostly prevalent. So remaining questions to be answered are, how is this going to play out in terms of epidemiology? Would this have an implication for metastasis, which ultimately is one of the reasons why the devils die? What does this mean for the immune evasion of these cells? Well, if these are questions you're interested in, I highly encourage you to follow our work at the Wild Genes Group. And I want to thank my wonderful lab and all the funding agencies. And I'm looking for jobs.
Okay, thanks so much for coming, everyone. And thanks especially to the organizers for putting together a really energizing symposium today. So we've heard a lot about cooperation and conflict in organisms kind of across life on Earth. And my argument to you today is going to be that viruses are also organisms that we can think about in this context. So first things first, what am I talking about when I say viral sociality? What do I mean? So the key to viral sociality is to zoom in and think about really what viruses are and what they're doing. So there's two things that all viruses are trying to do. They need to replicate their genomes, and they need to package those genomes inside capsids to go and infect new host cells. And the key thing about both of these processes are that the proteins that are required to do them can typically be shared within a cell. So replicase enzymes that one viral genome produces can typically replicate other viral genomes inside that cell, even ones that didn't encode them. Now, this means we can think of them as public goods, and I'm sure in this symposium it's no surprise that public goods are vulnerable to exploitation by cheats. And the way that cheating happens in viruses is super, super simple. If you're a virus and you delete the gene or the part of your genome that encodes for a public good, that makes you shorter. Being shorter then gives you a replication advantage. And these advantages can be really, really big within cells. On a single cell level, they can be 10,000 to 1 cheat to cooperator genomes. So because of this, we've kind of seen these guys historically kind of all the time in virology. So these are the very first tissue culture studies ever done on influenza. And this guy, Preben von Magnus, was seeing cycles. These cycles are caused by the periodic accumulation of cheat genomes that crash the populations. And we now see this in kind of almost universally. Whenever you try and grow viruses in the lab, we can now look at it in kind of more detailed genetic information. But here what we're seeing is we're seeing the invasion of cheat genomes that have deleted the public good parts of the viral genome. So the fact that these guys are kind of seemingly so ubiquitous in the lab raises this question for natural infections. And this quote comes from the fantastic review article in 1970 that kind of defines these entities in the viral literature. And there's this idea that if these guys crop up in the lab, maybe viral infections are kind of intrinsically self-limiting. Maybe this idea of social conflict is kind of inevitable and will drive viral infections extinct. So this raises this question. If it's happening in the lab, is it also happening in nature? And it's actually quite easy in principle to try to find this out nowadays because we have a lot of sequencing data. So if all that viral cheats are big deletions in public goods genes, well, we can just go and look for that now in all these mountains of sequencing data that are coming out. So the first thing I'm going to show you is my attempt to do this. And technically what we're doing is we're drawing reads from the viral population, mapping them to the genome, and we're finding these big deletions that are skipping over the kind of public good part of the gene. And this has been well characterized for lab systems. My collaborator Andrew Rao has done a great job there. And here we're going to look in 200 patients that are naturally infected by influenza, data coming from the New York State Department of Public Health. So I'm going to skip through a whole load of bioinformatics here, and I'm going to give you just the headline result. On the x-axis, we're going to go through every patient in this population. And on the y-axis, I'm going to plot 1 point per patient. That point is going to be the relative abundance within the viral population of these deletion mutants that have deleted the public good gene. And that point is going to be the most abundant cheat genotype in each patient. Here's what we see. We see this really wild result where not only are they there, but they seem to be there in numbers. So in 1 in 3 patients, the cheat genotype that has knocked out the coding part of the public good gene is at more than 50% abundance. It's the majority variant. So there's more to do here empirically, but this key result that they seem to be not just an artifact of tissue culture, but actually something that also plagues natural viral infections, is now going to motivate the kind of second part of my talk, where I'm going to talk about trying to explore the evolutionary consequences of this conflict in viruses, in the same kind of framework we've been hearing about today. Okay, so they're there. What you really want to know is, what are they doing? So the model I'm going to present to you now is really set up to produce outcomes that look like the data we can measure. So the data is we can detect these large deletions, we can measure their abundance. So we have these kind of deletion abundance plots that we can generate, not just from flu, but potentially from any virus that we have deep sequencing data for. When I say we, this modeling is done in collaboration with 3 fantastic collaborators, orchestrated by the Santa Fe Institute. So anything that looks kind of difficult and technical, these guys did, not me. So if you like equations, I've put one up there at the top for you. This is a stochastic birth-death model. If you don't like equations, the key thing about this model is we're tracking the population abundance of every possible deletion of the viral genome in the population in this blurry vector here in the middle. And we're going to do 3 things to these viral genomes. The first thing is we allow mutation to take place. And we have a model for this, and it's actually quite nicely behaved. So we have some nice analytics for this. But the most important thing for this talk is we allow sequential deletions. So you can get shorter, but you can't get longer. Second thing we do is we shake this vector, and we see what falls out. So technically, this is a Gaussian white noise approximation. final thing we do is we go through this population and we allow growth to occur within the viral genomes. And this is the interesting part where selection starts to come in. So everybody can grow if you're a viral genome. If you're a full-length genome, we assume that you produce the public good that's required, and so the whole population is essentially constrained by the population of these cooperators. The important thing we kind of enter into this is we say if you've deleted a gene, you can't infect a cell on your own, so you have a kind of density-dependent growth rate depending on the wild-type cooperators. And the key thing we put in here is this alpha term. So we're saying if you get shorter, you can be replicated more quickly, and we tune that with alpha. And you can think of this like a composite parameter. It's kind of like relatedness in a social evolution model. Multiple things are going into this, things like the co-infection rate, things like the kind of biophysical relationship between speed and length within the cell. But this is kind of an open parameter that's going to tell us how readily can cheats kind of spread within this population. Okay, so to explore how this model behaves, we're going to pick parameters for the birth rate, lambda, and the death rate, mu, and we're going to kind of fix them all to make it look like influenza during days 0 to 3 of an infection, the acute period. And then we're going to vary alpha and see what happens as we kind of allow cheating to take these different importances. And in this plot that you're going to see, on the x-axis we've got time, and I'm going to fill it in with colors, which is going to be the abundance of different deletion mutants, whereby blue is cooperators that are encoding the public goods, and darker shades of red are increasingly large deletions that have knocked out more and more of the gene. So we see these kind of like these kind of invasion dynamics, which are fairly typical of these cooperator cheat models. So cheats invade, they do better if they're shorter, and we get a negative frequency-dependent effect such that cheats do best when they're rare in the population. And what's happening here is actually a selective sweeps on clonal cheat lineages, if we look at the absolute abundances on the right. And this broadly looks like the patterns we see from the lab data, where you get sequential invasion of cheats that are kind of longer and longer. Okay, so there's a lot that we can do with this kind of approach, there's a lot that we might be interested in. I'm going to focus in for the rest of this talk really on this question of when do we get cheat-driven extinction. I think this is the big question from a kind of social perspective. And we're going to start off by using this model to get us some qualitative predictions on when that might happen. And the key to this is I've just shown you a plot with a particular advantage of cheating, where the cheats kind of invade and they reach some, the cooperators persist, but the cheats are kind of abundant within the population. If we rerun the same parameter values and the same initial conditions, we sometimes get extinction. It's a stochastic model. And the question seems to be, do you get a cheat that's good enough, early enough in the infection for us to see extinction within the 3 days? And I think partly what's happening here is you're essentially getting clonal interference between the different cheat genotypes kind of early on. So the way that we can explore this question, if it's self-limiting, I don't think this necessarily comes down to the kind of deterministic equilibrium. I think it comes down to this more combination of stochastic processes that we see playing out in the acute period of the infection. So I don't have analytics yet, but to kind of explore these ideas, I'm going to show you some numerical simulation values. Well, what we've done is we've run this model, we've solved it numerically, and we've varied key parameters that we care about. And the next slide, the conclusions all come down to these kind of heat maps, where in the dark regions, the simulation frequently leads to extinction of the population, and in the lighter regions, the cooperators persist. And there's 3 qualitative outcomes that I want to kind of highlight. So we can explore the importance of noise, this kind of inherent stochasticity in the process. And if you crank up noise, you initially start to get higher rates of cheat extinction, but it seems to be a little bit non-monotonic. And I think what's happening is if the infection is too noisy, you actually stop this kind of selective sweeping of the cheats through the population. We can also look at mutation rate, and not just rate, but also nature of it. So we've got a model where the mutations are happening, but also the kind of average deletion size is something that changes. And what we find is that the rate at which deletions happen in the population matters, the size of them doesn't. So we can start to make these qualitative predictions about kind of viral life history traits that might affect this cheat extinction phenotype. But the final thing is that really the most important thing is this alpha parameter, this question of how well are cheats able to spread in a viral population. And so the next thing I want to do in this model, and this next slide is really very much a work in progress, is I want to use the fact that we're explicitly modeling the data that we can observe to try to draw quantitative inferences from natural data sets. So in particular, we can simulate these dynamics under different assumptions about the cheat advantage and other parameters that we care about. We can use these to kind of draw out population-level statistics that we can measure about things like diversity of genomes in the population. My idea is that we can then use these to kind of infer from signatures from real data sets what might be going on. And in particular, there are kind of two things that I'm really interested in doing. So the first thing is I think we can infer this alpha parameter, this magnitude of cheat advantage, from natural infections. And the analogy here is really to the signatures of selection that we have from population genetics, where we can kind of statistically test if this alpha is important or not. The second thing that I'm interested in is I think these models can tell us what should we be measuring about these populations at early time points to predict the longer-term behavior of the system. In other words, can we take someone with the flu, can we measure the cheat population at an early time point, and can we use that to predict how severe the infection is going to be? So, in terms of take-home messages, the first thing is I think that sociality is not just a thing that viruses can do, but it's a thing that they have to do. It's intrinsic to the way that they replicate. What this means is that viral infections are plagued by the kinds of social conflicts that we've been seeing again and again in the symposium throughout the world. And I think what I'm trying to do here is take this idea of conflict, and it's not just a conceptual analogy. I think this can actually help us to formalize these things that we care about. So hopefully I've managed to convince you that it can be at least interesting to think about viruses from this perspective. And many thanks to many collaborators on this project. The modeling is done by my collaborators at Santa Fe. The data comes from the Wadsworth Center in New York State. Many thanks to Turn Lab and to Paul. And then finally, if you're interested in this stuff, I'm opening my lab next year at UBC, so please do get in touch. Thank you.
Thanks, everybody, for being here. I do want to start with some gratitude. First, thanks to the organizers. I once organized a one-day meeting, and it nearly killed me, so I can imagine how much work it is that we all rely on. I'd also just like to thank all of my colleagues for an amazing meeting, and I'd just like to highlight the trainees. Being a trainee is a psychologically challenging endeavor, even in the best of cases. And many trainees labor under other cases, and it's just amazing the work that people put in. I'm humbled. Turning to my work, I want to thank the larger community also for all the work that goes into developing the models and the mechanisms that I'm going to talk about today. My work is synthetic and tertiary, and it's just not even conceivable without the work of so many people, and so I'm again humbled. So following on that, some of you are specialists in the stuff that I'm going to non-specialistly talk about today. If there are any inaccuracies or precursors or things I've missed, I'll be in your deep debt. I promise that's my last debt. If you email me at scott@roy@sfsu.edu. So here's a summary of my talk today. I'm going to say that we have two major ways of explaining changes, or basically anything that we see in biological reality. One of those is adaptation, and the other is for seemingly non-adaptive cases. We sometimes wave our hands, but in most cases say drift. And I'm going to argue that in some important cases that drift hypotheses do not have strong theoretical or empirical support, and I'm going to instead suggest that we need to consider more the action of selfish elements and give some examples where I think they can be useful. Okay, so let's start out with a case study. I started my career working on introns, to make sure we're all on board here. Spliceosomal introns are non-coding DNA sequences that are in the middle of genes that get removed from RNAs. They're ubiquitous in eukaryotes, and there's huge variations in intron numbers across species. Most are not probably adaptive, though there are wonderful ways in which introns can be adaptive. They seem to apply to small minorities of introns in most species. Moreover, ancient introns have been lost en masse many times. So it's hard to explain this in terms of they're all there because they're adaptive. So an obvious place to go with this is a nearly neutral hypothesis. So if introns are slightly deleterious, which seems reasonable, then we would expect that intron insertions can fix when NE is small, but not when NE is large. Makes a lot of sense. Whatever one thinks of the specific models that have been put forward towards this, I would argue that those two assumptions are kind of hard to avoid. They're very likely. And yet, we see various things that don't fit well with this model. So there is no intron gain in many different lineages. Most dramatically, and importantly, for those of us who want to explain our genomes, there is no intron gain in conserved regions in 500 million years of vertebrate evolution in many different lineages. And that's been shown by many different groups. We see a positive and not a negative correlation between intron loss and gain rates, which doesn't make sense with differences in selective strength or efficiency across lineages. Multicellular lineages tend to mirror their unicellular relatives or reconstructed ancestors in their intron densities. So the presumed decrease in NE that goes with multicellularity does not lead to an increase in intron numbers. And introns appear to have been hyperabundant in early eukaryotes, not a famously small NE lineage. Okay, we can continue on to genome size, just looking at the size of genomes. Again, genome expansions being slightly deleterious seems likely. Again, we'd expect a correlation with NE. And yet, we found earlier no correlation between genome size and census population size in mammals. There's a brand new paper on bioRxiv showing no correlation between genome size and a proxy for NE in animals. And hopefully people saw Sarah Miller's talk yesterday showing no correlation with eusociality, and again, the presumed decrease in NE. It's worth pointing out how remarkable of a failure this is, right? So the general model in which we say that NE and selection on slightly deleterious variation is explaining it is the major explanator of differences that we see across lineages. It's hard to reconcile with the inability to find any effect here. I mean, I'm no fan of these models, and yet I'm shocked that there's no explanatory power here. Okay, so what is going on if that's not what's going on? So it definitely seems that selfish elements, and in particular TEs, so maybe I should say putatively selfish elements, play a big role. So whenever we see an expansion of massive expansion of introns, it seems to be driven by TEs that have acquired splicing signals that the spliceosome can recognize. TEs seem to explain most of differences in genome size, at least across animals. And I would argue that maybe less appreciated is that it could also help to explain broader phylogenetic patterns. Okay, so in general, this goes back to the classic theoretical work of Donal Hickey. We expect that TE's tendency to proliferate is going to co-evolve with the recombination rate. If recombination rate is low, then TEs are stuck with any mess that they make elsewhere in the genome. Whereas if there's a lot of recombination, you can take a chance, right? You can proliferate elsewhere in the genome and you'll be recombined away soon enough. And so then it's of note that meiosis appears to have evolved early in eukaryotes, consistent with the spread of introns, something that Donal Hickey said 40 years ago and then too many of us have ignored. But I think another one that's worth considering, and this is very preliminary data, is whether this can explain the observed proliferation of stuff or expansion of genomes in multicellular lineages. Genomes expand in some, but not all multicellular lineages. Multicellularity is associated with an increase in sex in some, but not all lineages. And of course, I know this data looks silly, and I agree. This is the level at which it is right now. But so far, it seems like it is those lineages in which sex is much more frequent that you see expansions of genome sizes. Okay, so to sum that up, I believe that there's remarkably little evidence for nearly neutral models of genome complexity and size, and that putatively selfish elements appear to be playing important roles. Okay, so now I want to talk about some other problems moving outside of the genome to phenotype and the kinds of answers that we can have. So here's a diversity of systems to give you just a flavor of the kind of stuff that keeps me up at night that I feel like we have terrible explanations for and that I don't think we can explain by neutrality or standard adaptation. So paternal genome elimination is a phenomenon in which males discard their paternally inherited genome during or before spermatogenesis. Another system involves the exosystem of Lobius, in which two different lineages have evolved the following. Males and females are both XO, so gametes either have an X or they have no sex chromosome, and then half of embryos die. This evolved from a standard mammalian system. If you have an adaptive explanation for it, let me know. We see ZW sex-reversed males in tongue sole, in which a mutation in the sex-determining Z-linked gene has become dominant, giving XZW males and skewing the sex ratio and frustrating the poor people who are trying to sell the larger females. There's a brand new paper showing heterozygosity requirement in a certain butterfly. So butterflies ancestrally have a Z-linked masculinizing gene. But in this lineage, not only do you need to be ZZ, but you need to be heterozygous for this. This leads to low fitness production of low fitness ZZ females. Again, hard for me to explain by neutrality or by adaptation. And finally, we have doubly uniparental inheritance of mitochondria, in which females are normalish, whereas males have a combination of their paternal and maternal genomes and transmit only their paternally inherited mitochondria. I want to point out that while neutrality is often invoked, reflexively or otherwise, to explain things, that it's really hard to get anywhere with neutrality here. If we think about the phenomena I just talked about, many of them are likely to have evolved through a step that involves a sex ratio skew. Sex ratio skews drive extremely strong selection. So the mean individual fitness for males and females can be written down as so, the difference can be written down as so, and so we can try to translate that into how much of a skew drives this sort of threshold, nearly neutral 1/S phenomenon, and that's the number. Okay? So a fraction of a % of skew is strong enough to drive a very strong selective coefficient for sex reversals. So for my way of looking at this thing, this excludes near neutrality as useful in explaining these things. So hopefully I'm going to offer you something and not just throw tomatoes at others. I need to put myself up as a target for tomato throwing. So I would suggest, what I want to suggest is that selfishness can drive evolution. It's obviously inspired by Arlen's famous 1999 paper. So when we often talk about selfishness, we talk about kind of manipulation by a selfish element and suppression. So either X drive and suppression of X drive, or mitochondria abortion of pollen and suppression thereof, or more generally readjustment. So maybe one gene drives an increase in fetus size, and other genes will then drive a decrease, perhaps by different mechanisms. But in all these cases, we're talking about sort of the return to the status quo. But of course, other possibilities are possible, and I want to point out 3 of these. So one is that an unsuppressed selfish element can drive a major change that then can be stabilized, but basically it's just a one-move thing. The other possibility is that selfish elements, by their definition, impose fitness costs on everybody else, and that these fitness costs can lead to selection for further adaptations. That'd be my number 2 down there at the bottom. And that the third is that selfishness can stabilize things that we think about as the status quo and haven't even thought about a selfish contribution to. This is hardly a new idea. Here's a classic case, turnover of sexual systems in angiosperms, and recently it was found in a snail. So angiosperms are ancestrally co-sexual, with an individual having both male and female functions. But you see again and again that mitochondrial DNA has evolved to thwart pollen production, presumably to redirect resources towards ovule production. So that would be sort of a one-step model, getting to females and co-sexuals, which you see many populations in. But then you sometimes have further evolution with the shortage of pollen driving evolution of male-determining alleles, leading to separate sexes. Okay, so let's talk about, I'm going to hopefully talk about 5 ways in which I think it's worth considering selfish genes. So we'll start with one case of the kind of single-step model. Okay, so the evolution of self-fertilization in plants from the ancestral case of self-incompatibility is generally assumed to be adaptive due to changes in ecology, mate availability, and so forth. So here's a quick tutorial in the mechanism. Self-incompatibility is encoded by a single locus that has 2 genes, one expressed on the pollen, one expressed on the ovule. The different alleles have a lock and key mechanism, such that if the pollen and the ovule it lands on share an allele, that mating is rejected, whereas if they do not share an allele, that mating is accepted. In many different lineages, you see loss of self-incompatibility through specifically loss of the pollen gene. Okay, And so I want to argue that this could be, that the alleles, I want to focus on these mutant alleles that knock out that function and ask whether, in fact, perhaps they are selfish, not responding to, at least solely, to standard ecological pressures. Okay, so what consequences do we expect of these? So one is we expect lower fitness of the subset of the seeds that are selfed or due to selfing. We expect greater relatedness to seeds. The mother transmits 2 alleles, not 1 to the seed. And we expect greater male fitness. And the reason for that, diagrammed at the top, a non-mutant cannot fertilize some ovules in the population, whereas a mutant may be welcomed by all ovules in the population. Okay, so we can depict this as that, as so. On the left, we have the wild type, and then next to it, we have the sort of components broken down. And what I want to highlight here, so in the middle, then, we have, this would be a mutant plant that's heterozygous, that can self because it's heterozygous for a selfing allele. And so there we're talking about the fitness of some allele from some locus in the plant, not the SI locus. And then the third column is the mutant SI locus. And there are 2 things that are unavoidable here. One is it has a greater male advantage because all the pollen that carry it have reduced rejection. And the other is that it has greater relatedness to the self seeds because all the pollen that is accepted carries it. So what that means is that it is unavoidable, as far as I can tell, that the mutant allele is expected to have greater fitness than the plant itself. Now, the fact that incompatibility is maintained is interpreted as that left column generally being larger, perhaps much larger than the second column. So we can sort of visually imagine that by that the left column starts really big and it fluctuates around, whereas the second and third column stay sort of in the same point. As it fluctuates around and it happens to come down due to ecological changes, it's going to hit the third column before it hits the second column because it's closer, it's higher. And so what that means is that it's easy to imagine what I've depicted here, a case in which the wild type fitness is lower than the mutant allele but still higher than the plant. So that means that basically the allele gets an advantage but the plant carrying the mutant allele gets a disadvantage, which is the definition of selfishness. So I've argued that self-compatibility could be evolving due to selfish alleles. Let's move on to another case of multi-step adaptation, which is the question of why does haplodiploidy evolve? So haplodiploidy, I'm here defining as instances in which males only transmit their paternal genome. On the right, you can see there are a few of these. Either males can develop from unfertilized eggs, or they can have a paternal genome but silence it, or they can have a paternal genome, express it, but not transmit it. These systems evolved at least 10, perhaps 20, maybe more times. And we can talk about the evolutionary dynamics of them. On the one hand, it's a great idea. It allows for maternal control of sex ratios, virgin queen or virgin female reproduction, and so forth. But on the other hand, when it first evolves, and this is a binary evolutionary change, we expect a genome-wide uncovering of recessive deleterious variation, at least in most forms. And we expect developmental costs. So we actually expect these new haploid males to be quite crappy. Okay, so how can we get at what drives the evolution of haplodiploidy? Well, the one major observation we've known for a long time is that haplodiploidy tends to evolve from XO-XX systems, where males are XO. And it seems to maybe particularly be the case when the X is gene-rich, when the X is large. The classic explanation of this comes from Hartle and Brown, which say that X-linked genes purge recessive deleterious alleles, reducing the cost of haplodiploidy. The problem I see with this is that the X chromosome is only often a small fraction of the genome, and so it seems like that would be a small benefit and unlikely to be the one thing that finally tips us over. But how can we test it? Well, we have an alternative explanation, and that's that X chromosomes, by their nature to drive, can cause a deficit of males. And so when there's a deficit of males, males are extremely fit even if they're crappy. And so that basically male limitation due to X chromosome drive could select for alternative means of producing males, even crappy ones. We realize this could be distinguished by looking at the form in which the paternal genome is present and expressed but not transmitted. So here we have a new mechanism of producing males that does not involve uncovering a recessive deleterious alleles. So long story short, my excellent student Noelle Anderson looked at this in 3 different origins of this kind of system and found remarkably large X chromosomes. I'm not sure if people can see this, but here the X chromosome accounts for about 1/3 of genes, here even more than that, and here also about 1/3. So, though not conclusive, this is consistent with the notion that male haploidy is evolving when you have large X chromosomes, regardless of the recessive deleterious allele, consistent with X chromosome drive driving these turnovers. And if I can further speculate yet further, I think it's interesting to think about whether other means of reproduction that don't rely on standard male production could also respond to these pressures. So it was just reported that Caenorhabditis, that the separate sexes of Caenorhabditis, those species often have female biased ratios, and there's a large X chromosome in Caenorhabditis. So that's a speculation, or perhaps asexuality in females to ensure reproduction. Okay, so the next is the craziest part of my talk. But in part, I chose to include it because I want to show you how crazy these things can get and how many transformations you can have. So this is the M. orgoni system, the creeping vole. We published its genome a couple of years ago. This has lots of crazy stuff going on. So let me introduce that the mammalian X and Y system is extraordinarily ancient and has changed only by occasional autosomal fusion. Has been very stable. And then in this one lineage, there are no intermediates. It's just this one vole that looks like all the other voles to me. The X is fused to the Y. Then the X is fused to the Y again. There's non-Mendelian inheritance of both of these Frankenstein chromosomes. Males are XX and females are XO, so that's reversed, and exist and is also reversed. Non-recombining, and the X chromosomes are non-recombining. So I'm not going to say this model is plausible. In general, I'm adopting the model here that a slightly implausible model, or even more than that, is better than no model at all or just hand-waving. You're welcome to disagree, but that is what I'm going with. So here's a set of steps. I'm going to go through this uncomfortably fast. If you want to see, it's in the bottom right corner. So feminizing X chromosomes are X chromosomes that cause XY individuals to develop as females. We see them in a variety of different rodents. They get an advantage because YY individuals are inviable. This could drive a Y to fuse with an X because basically X's are losing fitness because they Ys are losing fitness because they don't have X-linked genes. This is then expected to leave with problems with Y chromosomal expression because in XX individuals, it's going to be silent some of the time, which could potentially lead to another fusion or transformation into a non-masculinizing XY fusion. But now you've got fertility problem of XX males. XX males generally have fertility problems. This could drive the loss of one X during spermatogenesis, as we see. But that's then going to lead to XO females. XO females in studied mammals have fertility problems, which could lead to the driving of the X to restore the XX karyotype during oogenesis. So I think what we can conclude from that is that I'm really in on this stepwise model of things. Truth in advertising, right? Okay, so I'd like to talk about 2 more things. The first is, and both of them involve the possibility that selfish elements are stabilizing what we think of as the status quo. Okay, so gynogenesis is a form of asexual reproduction that's seen in various lineages. It's the form in which asexual females need sperm to reproduce. The sperm do not transmit any chromosomes, it's asexual, but they are needed to stimulate reproduction. So often these individuals will mate with males from other species, but what about when other males are not available? So I'm sorry, I should have cited various work here. I apologize to the authors. So there's been some really nice work done in gynogenetic hexaploid carp in which there's a masculinizing B chromosome. So basically, males are in large part caused by the presence of a masculinizing B chromosome that somehow makes it into the embryo despite all the other male's chromosomes not making it. And so I've shown that here, that basically you can see that the M makes it into the offspring, but the other red chromosomes do not. So note here that the presence of this masculinizing element is a real bummer for the autosomes, because by virtue of the action of the the masculinizing factor, that individual, instead of being an asexual female, is a male that will never transmit its chromosomes. And so this is kind of an ultimate selfish genetic element, as Warren and Stautheimer described the PSR chromosome in Nasonia. Moreover, you see in these males that carry this B chromosome leaky autosome transmission, leading to some recombination, possibly helping to avoid mutational meltdown and stabilizing the system in a second way. Okay, and so finally I'd like to talk about maintenance and loss of sexuality. The well-known paradox of sex is called the twofold cost of sex, whereas asexual offspring inherit twice as many alleles from their parent. And so a female engaging in sexual reproduction is giving up half of her transmission. There's a huge amount of work here which I'm not qualified or given the time to summarize, and I don't mean to give it short shift. But it seems like one part of this explanation may be loss of heterozygosity. So here I've depicted the two major ways that we see asexuality evolving. So we have meiosis 1 and then meiosis 2, giving us those 4 products there at the bottom. And then we see the two major ways that you see asexuality deep diploidy restored is either fusion of the two terminal products, we'll call it terminal fusion, or fusion of the central one, we'll call it central fusion. And then, as you can see here, in either of those cases, in standard meiosis, you get substantial loss of heterozygosity, uncovering recessive deleterious alleles, and presumably leading to these individuals often having lowered fitness, and helping to explain the lack of asexuality, or the persistence of sex. However, there's another form of meiosis which has evolved many different times in various eukaryotes. And in that case, we have a different situation. So here, the centromere breaks in the first divide, and so we can just follow this side here. So what happens now is recombination occurs during meiosis 2, and what that means is we have reciprocal products here. So when these fuse, you get no loss of heterozygosity. But the paradox is that these systems seem stable. We have whole groups of these, of lineages with inverted meiosis that have not succumbed to asexuality and meltdown. And the one thing I want to point out here is that if we think about these individuals as not cells of the mother, but as having their own selfish reproduction, this selfish interest, we see that this individual has no twofold cost. Because this individual shares no alleles with its potential fusion partner, this individual gets no benefit in terms of transmission from fusing here. So if there's any cost to sex, and any possibility of this individual refusing the fusion, if it has its own volition, then we can imagine that this individual would refuse, that this product would refuse this fusion, and could, and thus selfish interests of meiotic products could be part of the maintenance of sex in these systems. And I just want to point out one more thing. So in those cases where we do see asexual evolution, in standard meiosis, we generally see it through central fusion, and we generally see it when there's terminalized recombination. So that recombination is only happening at the tips, so that through most of the genome there's no loss of heterozygosity. And I want to point out that under those circumstances, these little tips here actually have a very strong incentive to fuse, because if we have basic randomization of alleles at the tips, this allele here knows that it is present 100% here, and there's a 1/3 chance it's there, and a 1/3 chance it's there. So if it doesn't drive asexuality, it has a 1/3 chance of being transmitted into the sexual organism. But if it drives the fusion, it has a 4/3, it'll have an average of a 4/3 copy number. So in fact, this product has a 4-fold cost of sex, potentially explaining being involved in the driving of asexual reproduction in these cases. So here are my conclusions and assertions. In general, whether you buy these specific models, my main goal today has been to say that we hope to convince you that selfish elements could be important and that we should consider them and that we need them because our neutral models are not doing as much as we want them to. I want to acknowledge my lab, including about 13 new undergraduates who are looking for sex determination loci in Hymenoptera, which is really exciting. Various collaborators, funding, and again, everyone for letting me know what I've missed here. I'll put my conclusions back up and thank everybody for a great meeting.